Jesús, ¿me escuchas? Alonso, ¿me escuchas? Alonso.
Yo te escucho, Luis. Perfecto.
Sí, ¿están en silencio en el auditorio o es que me parece?
No sé.
Reportando sintonía.
Listo.
Listo, Alonso.
Silenciemos micrófono que ya empezamos a transmitir, entonces sigue Alonso entonces transmitiendo.
Listo, señor.
Listo. Entendido.
Un, dos, tres. Un, dos, tres. Un, dos, tres. Un, dos, tres. ¿Se escucha? Un, dos. No, es que ahí no tienen como, no sé, como capacidad de muy lejos, ¿no? Tuve casi pegarlo.
1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3.
Sí. ¿Don Jesús? ¿Diana?
Alonso, aquí escuchamos perfectamente, yo escucho perfectamente.
Ah, listo. ¿Diana me escucha? ¿Diana me escucha? ¿Ahí me oye ahí? Hábleme.
Alonso, yo creo que ya no ha desactivado el micrófono del jueves. Háblalo
Diana,
por red.
por favor, desactive
Sí,
el
te
micrófono.
escucho.
Listo. Por favor, ¿ahí me está escuchando? ¿Ahí?
Sí, sí, perfectamente.
Espere que voy a hacer varias pruebas. ¿Ahí me escucha?
Sí,
¿Ahí?
suavecito.
¿Ahí me escucha bien?
Sí,
Bueno. Haga el favor y desconecta el micrófono, por favor.
bueno.
Gracias. Ahí.
Okay. Uh-huh. Today we will continue with the second part of the talk of the Professor Marie-Louise Saboungi. He's coming from the University, the Pierre and Marie Curie in Paris. And we appreciate very much, Marie-Louise, you are coming, you are accepting our invitation to do this talk.
Thank you very much. And really, it's a real pleasure to be in Colombia and to be in Cali. And today I will proceed and go to the second part. Now, some of you have asked me a question yesterday and I haven't yet answered it, but I will, feel free to send me questions by email and I will answer them if you can't say them. Okay, so today I will be speaking about the second part, and it's kind of like a continuation, but it's a little bit different from what before. And you remember before we did them with polymers, we wrapped them, we did the iron oxide, nanoparticles, and today I will talk to you about another alternative, and this is confining these nanoparticles, exactly the same ones, in hydrogel. And so I will present to you the structured emulsions. And what we did is the production, the characterization, and then the validation as a thermomagnetic delivery system. And okay, so what are the challenges? It has to be a bit different from what we did yesterday. So in this case, what we're going to study is exactly a different challenge. We would like to design devices for long-term therapies, okay, with a zero premature release rate. And this is what's called in the literature kind of like the on-demand release. So kind of like you push a button, you have something coming out, you push, you stop, and you keep going, okay? And so there is on and off mode. With the on mode, what you do is it could be activated with a controlled release rate, but when you put off, you want to make sure that there is absolutely nothing which comes out. Now, usually it's very hard to achieve a completely off mode, so we'll be happy if there is a continuous leakage, but it should be negligible, so very little, if any. And so a too rapid release or burst release many times can lead to very drastic side effects. So we would like to avoid it in this case. And so what here I'll be presenting to you is a choice of a combination of drug, like we did last time. And with this drug, it's carrying a lipid particles, and they have a very high surface areas, and they have a stimulus responsive hydrogels, and these hydrogels are in a way replacing the polymers that I showed you yesterday, which was wrapping around these drugs. In this case, what we used is a hydrogel. So it's also, of course, biocompatible. And this approach, we feel that it's a particularly promising. So what is the problem? Well, this, in a way, what we would like to do is create a material which is biocompatible, and it contains a drug and a hydrogel, and we can control the external diffusion by external stimulation. Okay, so we're using the same magnetic properties, and we're going to do it through a control. So what we use now is isosomes, which is like a colloidal suspensions, which is made of water or oil, and this is used as a container. And we use the magnetic nanoparticle as a thermomagnetic activator. So we're again relying on the magnetic properties of these nanoparticles to ensure a delivery and an activation. And here what I'd like to present is also we used different techniques than in the previous work. And so we have a remote thermal activation by an alternating magnetic field. And we monitor the drug diffusion by EPR or electron spin spectroscopy. So, well, for those who like to see things, this is the visual appearances of what we came up with. The black dots are the magnetic nanoparticles, and again, they are the iron oxide, and we synthesize them in the same way like in the previous work, but instead of the polymers and the big mass blue is the isosomes with the drug in it, and the drugs are also in red. And what we use in this drug, what we used is well, I'll say it in the next one. And with the hydrogel, it's a kappa carrageenan hydrogel. And we use the isosomes that you see in blue as containers for the drug. And this is here I put the formula of the kappa carrageenan. And so what we have, we form a physical gel at about 20 grams per liter, and this could work at room temperature. Next one. And here is a small, I think we buy it. Yeah, it's available. And it's a good question. I don't know if we had to do any supplemental things to it. I don't think so, but it's available. You can buy it. And this is a small reminder of the difference between the magnetic properties of these iron oxide nanoparticles. So here we have the multi-domain, and as you can remember, we have the hysteresis loops, and we have different properties. And on the monodomain nanoparticle, or what I would like you to remember is this very steep curve around 0. You see how the slope is very strong, and this is the indication of the magnetic susceptibility of these nanoparticles, which will be very useful later on when we put them in a magnetic field. Okay, and in this case we use electron paramagnetic resonance, or ESR, electron spin resonance. It's kind of the same thing. And we use it because we could detect with it both the magnetic nanoparticles and the drug with a high sensitivity. And we also explored the possibility of having in vitro and in vivo experiments. And in this case, the PhD candidate was very lucky, because she had to go actually to Switzerland and do these measurements and at Lausanne at the EPFL with our collaborators, because we don't have all the equipment. And well, here is a summary of the interesting results that she obtained there. And as you can see here in the first plot, well, all what it's showing is that the ESR spectroscopy showed that the ferromagnetic relaxation is visible. So you see here, there is this spot that we highlighted, and which is developed in the inset. And as you can see, we can obtain the signals of the magnetic nanoparticles, and on top of them, we have the signal related to the nitro oxide. And what is the most interesting results is the bottom two figures. So if you look at the one on the left hand right-hand side, what you see is you have the spectra, the intensity as a function of the magnetic field. And the one which is highlighted, well, this is the effect of the tempo, and you can see that we can see a difference in it. And we did in the next one on the right-hand side, what you have is two different results. One is when we had the tempo solubilized in water, and it's in blue, and in oil. And what we can see is really that it doesn't make any difference if we use the water or the oil to solubilize the tempo, and the tempo is the drug which is being used. And so, well, by looking at these figures, what's the conclusion that we can draw? Well, that we can find tempo quite clearly in the system, despite the fact that we have so many components. So the tempo is clearly visible. And the second thing, if you look at the red and the blue, well, there isn't such a difference in the location of these peaks. The magnitude, of course, changed, but the position are unaltered. So what does it mean? Well, it means that tempo is not trapped at the interfaces, so it moves, right? And this is what we wanted, is to be able to get out of the system, but it remains fully mobile, both in water and in oil. Okay, next one. Sorry, go back. Right? Sorry, in the first one? Yes. Well, sorry, you want to show where? Yes, yes. Oh, no, actually, we only saw it there, and it was kind of expected, so it wasn't any unusual result. Yeah, but it's a good question. Yeah. Okay, so what does this result show? Is also it shows that the less, since we had these. Sorry, David, can you go back? Since we said that we have this nitro oxide EPR signal, so if we now, please, David, the next one. Yes, so what it shows is that the lecithin plays a key role in controlling this diffusion, which is something we expected, but it is independent of the molecular details of the drug, and at least in the case of these small molecules. Well, the other thing is we didn't see anything, any effect of confinement. The tempo remains mobile, and if you put it in oil or water, it has the same mobility, and it's kind of really interesting and a bit exciting because it doesn't make any difference if it is trapped or it's not trapped. So anyway, it doesn't hamper the movement of the drug. Okay, having it with all the things around it. Now the next measurements, in this case, she also had to get out of Orléans, which is kind of nice. So we measured alternating magnetic field, and there is only one that we were able to localize in France because it's not something commonly used, and this was in Bordeaux in the equipe of Etienne Duguay. And this is kind of like the schematic with it. And this will help in measuring essentially the temperature rise in a sample when we apply an alternating magnetic field. Okay, Dave. Next. Well, actually, the sample. Well, yeah, the sample was, you know, of this size, so it's kind of macroscopic. It's not microscopic, so you could see it. Okay, and it's kind of it's a very nice device. And then when we went back to use it again, it was always not working. So we couldn't do further measurements. But let me tell you what happened. Well, in this case, this is what I mentioned about the on-demand release. And if you look first at the figure B, so what we do here is we apply, we raise the temperature, right? And this way we did it for almost 20 minutes, the first one. And then you let the temperature drop. And then you do it again. And this time we went like 40 minutes. And we went above 30 degrees, and then we let it come down, and then we have an ice bath, okay? So we only triggered it twice, right? So we pushed the button on, if you could imagine a button on, it went up for 20 minutes, then we off, and then on again, and then on off. And the total was about 60 minutes, and then the ice bath. And then up, what we could measure is the percentage of tempo release. And what happened is in this, and then what we have here in the figure A, we have two results. One where we have the percentage of tempo released, and then the second one, the red, we did like a blind test where we had a passive activation in a water bath. Okay, and as you can see here, there is, of course, the water bath is maintained at 37 degrees centigrade, which is kind of like, you know, it's a little bit higher than the last temperature. And what you can see here is that there is really no difference within the experimental error bars between the two results. So what this means, well, if you want to go back to the isosomes, well, when they are immobilized in this hydrogel, well, they play the role of both drug reservoir and, on the other hand, the drug delivery. And so they will allow this sustained release of the drug as a function of time. And the AMF triggered at 37C gave similar results. So the main effect of the AMF is a temperature effect without any burst release. So anyway, it's feasible to do what we were planning on doing. And we have the concentration of the released model drug was independent of the mechanism of heating, since we have in both cases the same results. So what did we achieve here? We were able to specific targeting of cancer cells. We did a selective and sensitive multimodal imaging, and we could control, anyway, the drug delivery. And this is targeted by, triggered by either targeted environment or by external control. Okay, so this work, anyway, what we did in it, and this lasted 3 years, is kind of like do this synthesis with the isosomes and put a drug and look at it, and then see that this system of drug release on and off is something which is possible with what we have. Okay, so then after this work, what we decided is what more to do. Okay, you can play more games with putting other drugs and do different cooking, different synthesis, and so on. But you know, again, what we want to do is understand the basic of the system and all the properties with it. So one of the things that we started doing is to look at the structural and magnetic properties of these magnetic nanoparticles. Okay, so as when you make such synthesis, you go to your lab and you use TEM or STM or something like that to measure the size of these nanoparticles. But there are other techniques, and there you have to take a specific amount sample, put it on how do you call it, on, right. And then when you're dropping these liquids, or what you do is, since they are sitting in a cavity, in a way they are being slightly confined. And what we noticed is if we do microscopic measurements, or we look with a small angle, X-ray or neutron scattering, well, there is a difference in the results between what we get from the microscopy and what you get from the neutron or the X-ray measurements, because in this case, you're taking a free sample without confining it on a grid, okay? So what we started doing is small angle neutron scattering and small angle X-ray scattering, and then we also were interested, there are two ones, sorry, the number 2 is measurements of the internal structure. So, to go back to the measurement of the small angle, you can do them by neutrons or X-ray, and each of these techniques will highlight different elements. So for example, if you're doing it with X-rays, you're going to see the heaviest element. So in the case of iron oxide, so you will see more of the iron than the oxygen. And if you do with neutrons, it could be the reverse. So you highlight different elements. And so we started doing and also you can do, and you can also check the blocking temperatures. The second is you can look at the internal structure of the magnetic nanoparticles. And it is very interesting because you can get the distribution, you can see how, you can get the morphology of these magnetic nanoparticles. And the third thing we wanted to look at is the influence of the shape of the nanoparticles, and I'll come back to that. And so we started this work at the Advanced Photon Source at Argonne, and the neutron studies in Berlin, and in ISIS, which is not the ISIS terrorist, it's the name of the neutron source in England. Okay, so this is an illustration of the small angle X-ray scattering. And as you can see, there is here on the right is the high resolution electron microscopy image, and on your left-hand side are the results of the intensity of the small angle X-ray scattering. And you see you can make fits, and from the fit you can get a size of the nanoparticle, and in this case we got 2 nanometer with a sigma equal 0.16. And what's also shown here is that you can also get, you see we have here for different samples, you can compare the results between the SACs and the TM. And what is always amazing is the difference. For example, in the first sample, one find by fitting the results of the small angle X-ray scattering a diameter of 2 nanometer. And when you use the TM, with a high resolution TM, you find a 3.2. And it goes on. This is not our work. This is work taken from Nunes and al. And as you can see, it's a little bit surprising that we have these differences. But again, we can explain it on the basis sometimes of the technique used and of the environment. This is a little bit you know, this confinement and some pressure exerted on the sample. Next figure. Now the pair distribution technique, this is, again, you can do it combining both X-rays and neutron diffraction because they see it in a different ways. And what is interesting is, in this case, this is the experimental X-ray diffraction structure functions. And you can see the differences between the distribution between the black curve and the experimental data, and some are calculated and the other one is measured. And what did they come to the conclusion that the properties of the iron oxide nanoparticles differ sensitively from those of the bulk materials, which is something one expects. And also, they can locate where these nanoparticle atoms reside. And in a way, they can see where they reside at the grain boundaries and where some of their neighbors are missing. And, you know, always having two techniques give you a better insight into the nanoparticle structure. Now, also, what is interesting is to look at the differences between the particle shape in all of the samples that we made. These iron oxide nanoparticles were spherical, right? And there was a distribution in the size and so on. But as I mentioned yesterday, they are now very different sizes and different ways of doing these nanoparticles. And I would like to here, this is illustrated by the work of, I don't know who, but yeah, by Rocca and al. What is interesting is now the same systems, the iron oxide, you can do them in nanorods, in nanorices, in nanocapsules with rod-like morphology. So there is all these changes, and we're working on it. And please, the next one. This is a paper which was just accepted in scientific reports. And what we did in this paper, this is our work. In this case, we did the hollow gamma iron oxide and nanoparticles. And this is quite an interesting thing because look here, you have the A gives you a typical high resolution TM. And now with the B, you can go further, increase the resolution, and you can have much more details about the nanoparticles. And now the bottom one is a very interesting one that I really wanted to show you. This is, as I mentioned yesterday, in these iron oxide, we have two valences of the iron usually, and we would like to detect how much of each we have. And in this case, in the past, we used this spectroscopy that I showed you yesterday to determine the quantities of the different forms of iron. And here, what we used is a very simple measurement. It takes like maybe 5 minutes maximum, not even. It takes more time to set it up, 3 minutes. At the synchrotron source, we did what's called XRDIND. And there you just look at the K edge of iron and you get the spectrum. And what you can see is the data and the fit. And so what we have up is the magamite and down the magnetite. And when we combine them and we fit them, we have a very nice fit, which is represented on top. And this contains about 75 or 80% of magamite and 20% of magnetite. And then you have the bottom one, the R factor is, of course, you know, the statistics of the fit. And as you can see, we can determine the amount of the valence of the iron. Now, why is this important? Well, no one has looked at it, and we don't know if we change the proportion, what would be the impact on the nanoparticles. Okay, sorry, yes. On the same, yes, on the same particles. So, you know, you produce a batch of them, and these are the hollow rods, so the inside is empty, and then you take the same samples that you used and you run them at the XPS, you run them at the APS, at the synchrotron, and you do, you know, you adjust the energy in a way that you're below the K edge of iron. And then you record this, and it really takes 5 minutes to do it. Oh, well, what we did here, this is the sample that we have, which is, it shows, oh, the technique is XRDIND, near edge X-ray absorption, near edge spectroscopy. So you stay near the edge of the element that you're studying, which is iron. And the experimental data are given here in red. So it's a continuous red. And then you take samples that you know of. So you have a sample which you know is 100% manganese. And so you put it here. And then a sample which is 100% magnetite. And you fit the data accordingly. And you see how much you need of each one to get the best fit. And so it's a standard. And again, it's quicker. And this, we did nanorods recently. And again, in this case, we did the X-ray diffraction, and we did the microscopy, and, you know, we get all the information. And we have done the XRDINDs, and we come up with proportions which are slightly different from the previous ones. Okay. And the other thing that would be in. Yes. On a foil. The nanorods are, you know, iron oxide nanorods, and we do them the usual, you know, in a suspension. Surface on a grid or something. Yeah, this is the usual. And, you know, there's two ways. You could either dry them, get them into a powder, or you could drop a couple of, right, carbon grid, right. Okay, so what else remains to be interesting to what are the variations that we could perform? Well, we could change the size, right? We can go a little bit larger. And in this case, we can have, this is work done by, well, it's down the, sorry, it's not appearing. This is in the literature. And what would be interesting is to have a monodomain, multi-domain size, and you can change them and change the size of these nanoparticles. And this will make them useful for hyperthermia, for example, the top one in the case of A and B. Or you can also do a different range. And in this case, you could address the problems related to the tomography imaging. Okay, and this is the large, little bit larger nanoparticles. Now this work, lots of it is, there is lots of this work that I'm citing here is done in a very famous lab in Brazil. And sorry, I forgot the names. So there is lots of activity on these nanoparticles. On these nanoparticles. going on. Yes, yes. It's very interesting and, you know, publishing really well. Next one. Now, the other thing is you can get out of the iron oxide, and here we have, for example, you can go to particles like cobalt, iron, gold, iron platinum with gold, and iron platinum with iron oxide dimers. So there is lots. The field is very rich. And these are illustration of different things. So in the case of A, you have a magnetic liposomes, and then arranged with dimers, iron platinum with iron oxide, and then you have a core shell silica. And then at the end is something very exciting because they fill carbon nanotubes with magnetic nanoparticles. And anyway, this is again studying the effect of confinement of these nanoparticles. And in this case, they chose carbon nanotubes for probably applications, I hope not related to health, because the toxicity of the carbon nanotube is always under question, except by those who work on carbon nanotubes, right? They say it's not toxic. But anyway, you could imagine confining it in titanium oxides or other biocompatible tubes. Next one. Okay. Oh, twice the same. Okay, and there is a special attention to the manganese perovskite, and you can see you have different relaxation time and so on. And I think this is the end. Okay, now I would like, all of this was on nanoparticles, and well, there is something quite exciting which was happening back at Argonne. By a team which works on magnetism and nanoparticles. So instead of working on nanoparticles, what they came up is with an alternative approach, and I would like to discuss it with you. It's a micro-fabricated magnetic structures. So what I liked in this paper that I will encourage you to read is called Nanomedicine. They have all these definitions of terms, and I just love reading it because it's very interesting, and it's differently viewed, right? So you have a bottom-up fabrication, and this is kept for a method for building large structure from smaller subunits. And then this is the second one is the top-down fabrication, and this is kind of like you know, fabrications which gives you magnetic structure, and you can fabricate it using top-down methods as opposed to chemically synthesized magnetic nanoparticles, which is the approach that we use. And what's the definition of the nanoparticle and the microfabrication? And so, given these definitions, let's see what's the next step. They go and they define the magnetic agents in the biomedicine. So you have on your left-hand side the chemically synthesized. So you see the supermagnetic beads, and these are interesting for hyperthermia, which we discussed, for MRI, gene delivery, cell separation. And then, well, what do they face it with? They face it with ferromagnetic disk. And this is very useful for magnetomechanical actuation of cancer cells, hyperthermia, so you find the hyperthermia, and the magnetic detection and the drug release. And then instead of using polymer encapsulated particles or nanowires with cell separation, well, they have in the microfabricated structures, they offer us synthetic antiferromagnetic particles, and you know, and they go through the method for detecting them. And then, you know, what is these microfabricated platforms and magnetic tags, and you can have magnetoresistance-based sensor and microfluidic chips. Now, what did they do? The next step is, this is the most beautiful thing because it shows you how a microdisk fabrication is done. You can go through the steps one by one, and the characterizations is done by SEM and TEM, and the step A, the step B. So you go from a silicon wafer, you spin coat it, then you use a mask development, and then you get magnetron sputtering, and you remove the mask, and all what you have is the disc-shaped magnetic particles. And the next thing, what is very interesting, is taking these ferromagnetic microdisc, and they used a spin vortex ground state, and having this disk and putting it in a magnetic field. And what they noticed is they need a very small magnetic field to get it to rotate. So when this disk is rotating, the magnetic disk oscillates, and then the next step, please. Yes, and here, what I think this moves if you put the thing on it. Oh, no. Yeah, it's here. So anyway, what they do is this is kind of like the scheme. So what you have is this microdisc that you put in a magnetic field, and this magnetic field is amazingly low. It's 90 Ørsted, and what you have down is all this structure where you have a drug inside a facing a membrane and all of this, and then here you have the cell, and when you put this magnetic field, this starts slowly rotating, and what happens, if we can go back, David, sorry. What happens is when this magnetic disk oscillates, what happens is the membrane integrity is broken. So you can see here all the assembly, and then when this integrity loss, you get a DNA damage, and then what you start is a programmation of a cell death, and this is due to the induction of these stretch-activated intracellular channels, and it increased the levels of calcium ions. So the next one, and as you can see in this AFM figure, you have the first one is the one before you apply the magnetic field. You can see the tumor, and you apply this magnetic field for a very short time, and what you get there is a destruction of this tumor. So this is kind of quite exciting, and actually it was published in Nature recently, and there is lots of comments about it. Okay. Oh, well, we can go back and look at the disk. David, can you go back? Oh, do they give the disk size? Where is it? Oh, sorry, it's down here. It's 1 micrometer. So it's kind of, from what described in the paper, the gene, what they are going after is brain cancer. And so it's kind of interesting. So what are the advantages of this technique compared to hyperthermia? Well, the advantage is that you have, it's transmitted directly to the targeted cell, which is also we do with hyperthermia, but anyway, it is transmitted directly with a very high specificity and a high efficiency, right? It all happens in a few seconds. And what is very exciting about it is the fact that the magnetic field applied is very weak. It's less than 100 Ørsted. And so it is 100,000 times smaller than used in hyperthermia treatment with magnetic nanoparticles. So you're really advancing enormously in getting away from having side effects or anything very important degradation of the cells. And so it's amazing that it could work. And they can scale it to smaller lateral dimensions, so of the order of 100 nanometer. And obviously, you know, there is much work which should be done. And well, this work that I presented to you dates from which year? Actually, 2000. Sorry. Yeah, 2010. But, you know, nothing yet has been published to my knowledge, but there is lots of work happening on this field. So it's kind of interesting to see how you can go a completely different way, but still the same principles. You know, instead of nanoparticles, you're using microdisk and still using the magnetic field, right, to trigger some movement. And this magnetic field is by far smaller than the one we use in the case of nanoparticles, which is a big advantage, but there is still much more work which needs to be done. Okay, and this is my favorite joke. Well, we all love Richard Feynman, right? He was quite an unusual man. And he said, well, what's the sequence? What is the sequence of bases in the DNA? What happens when you have a mutation? Where do the proteins sit? It's very easy to answer many of these fundamental biological questions. You just have to look. But the next one is a friend of mine, Albert Hibbs, I love that, suggests a very interesting possibility for relatively small machines. It would be very interesting if you could swallow the surgeon. So the surgeon can become a nano, right, and go inside and operate. Well, with this, I would like to thank my collaborators. Well, especially Karine Kaki and the team from Tours, and then Etienne Duguay and Stéphane Mornay for the use of their AMF. Irena Milosevic and Jovan Blanuza and Marin Tadic, they are from Belgrade in Serbia, and they were very essential in doing all the synthesis and all the characterization, and then many, many of the tests. And Laszlo Foro and his team for letting us use the ESR. And of course, the money which comes from to finance this project. And well, I'd like to thank you very much for your attention. Muchas gracias.
Podemos pasar a la sección de preguntas. Pueden preguntar en español o en inglés. Se puede traducir como se sientan más seguros. Ya, questions.
Buenas tardes. Yo tengo dos preguntas. La primera es que me gustaría saber qué emplearon como antifloculante o para impedir la aglomeración de las nanopartículas. Y la segunda es cuál era el método aquí en los discos para el rompimiento de la membrana, porque de todas formas eso es algo complicado, cancerígenas, dada la alta resistencia que presentan también al movimiento y a los medios.
Well, I'll start with the second one. Actually, Thank you for the translation. Yeah, the second one about the membranes. Well, it seems like this is, it's a very good question because I had the same, but then they tell me that it's, you know, this is what experimentally they got. It's kind of quite well documented in the paper. Now, to keep the nanoparticles separated, I honestly don't remember because it's, you know, you kind of give the technique and then it's being done and so on. I really don't know, but I could look it up. But it's something standard, it's whatever being used in synthesizing iron oxide nanoparticles, nothing unusual. But the only thing is, I think the solution has to be in an alkaline medium to help it. We kind of repeatedly made TM measurements because we had it in our lab and it's kind of free of charge for the people in the lab. And then what you could do is, you know, we repeated them at different times, like 1 month after we made them and things like this. And the sizes haven't changed since it, you know, we didn't notice any remarkable changes. But it's really a very standard technique, what we used. And yesterday there were questions about reducing the size, and I think this will be something very nice to do and trying to improve it to get a very nice size distribution, right, and not too much, you know, to get them as monodispersed as possible. Yeah.
Okay, I have the one part. No, follow. When I want to remark is that collaboration with hello. What I want to remark is that for the auditorium is that the collaboration in research is a very powerful tool. Yeah, that is the reason why we try to bring people like Marie and Dave. You can. Each of you can interact with the professors and get maybe some collaboration. Okay, that is the big idea. That is our goal.
Thank you.
Okay.
Actually, I forgot to put Argonne National Lab for the for the neutron, for the
neutron, the cyclotron,
right.
yeah.
Yes, but it's true, you know, science has no borders.
Okay.
Nice.
The other thing I want to remark is to bring Marie-Lou and Dave here is we have to thank very collaborators also. Collaborations de Consejo de Química, Consejo Profesional de Química. That was one part. The Faculty of Science, the Academic of Science also in Bogota, and the Department of Physics. I mean the graduate program in physics and in chemistry. So you can see a lot of organizations are support us for this opportunity.
Yeah, we hope that things work out.
And also aesthetics at the end.
Shouldn't forget that.
Oh, I do have a question.
Oh,
in the end. And this question came out when you were talking about how important it is to have mono-dispersed systems in order to have a really nice control of the system,
Yes.
especially when they get inside the body. But there was an image, and I guess that if some of my students of the surface chemistry class are around, they will probably notice that image that you showed some time ago. But those nanoparticles on a surface, and the nanoparticle is when it gets attached on a surface, then it spreads. So the original shape, maybe when the nanoparticle was made in a fluid, it could be, let's say, spherical, but when it gets attached to a solid surface, or when the particle interacts with the solid surface, then it may interact with the surface, so the shape changes.
Mm-hmm.
So in that case, when you put nanoparticles like this, let's pretend they are spherical at the very beginning in the original fluid, but once they are injected into the body and then they start passing through or passing, let's say, into the blood system, for instance,
Yes.
and then they get in contact with cells and all the parts of the body, then the shape may change and the control, it's probably becoming more difficult. Am I correct in this case?
Actually, you're absolutely right about it. And there is also, there is also the effect of, we kind of thought that if you put them in a nanotube, or if you put them in a tube, or, having these polymers around them,
Mm-hmm.
having all this dressing around them could help in maintaining the shape
Mm-hmm.
of the nanoparticles inside.
Mm-hmm.
But all of this remains to be seen, and I don't know what is the best way to check. But all what we know is what you put in, right? What goes in, and what's the effect that you see.
Mm-hmm.
But you're right. This is kind of like it's in its infancy,
Mm-hmm.
and we need absolutely to work in collaboration with people who do like more into biology, more into medicine, more who have this kind of knowledge that we lack. And this is why a collaboration with people like in the medicine or in pharmacy are very important, and mostly in medicine, because they have things that we never thought about. So kind of like crossing the borders is very important. And the other thing that I mentioned yesterday is the effect of the pressure on these nanoparticles, because when you're putting them also in the body, there is a slight effect of pressure, which is not, I don't know what, probably it's not very high pressure, but, it's still kind of like open to many suggestions and many ideas. It's kind of really difficult. And the simulations, they help and they don't help, because in a way, when you do numerical simulations, again, you're not looking at all of these things and you don't have the data. So it's a very good question. And actually, I never had this question before. Very interesting, yes.
Well, the whole thing is that in nanotechnology or nanoscience, people are looking for two kinds of control. One is the control of the dispersion, the
Right.
particle size, to
Right.
have just one specific size.
Right.
That's interesting, you can control many things. But at the same time, the control in shape, because the shape of the particle also dictates its behavior.
Yes, and this is why, in a way, we're looking, to answer partially your question, we're looking at different shapes. So we're going into the nanorods, into these hollow particles, nanoparticles, and the rice kinds, and so on. And I don't know what is the effect going to be. No one has yet tried it that I know of. So kind of like the field is open to many more questions.
Related to the Professor Walter question is that So the magnetic behavior is totally independent of the shape of the nanoparticle. Did you expect that, or it was?
Well, in a way, you know, it could depend on the shape, but you know, they are still, we're still in a domain where they are super paramagnetic, so we're taking into advantage this, and they have a very high susceptibility and so on, you know. So I don't know, no, we haven't yet looked, or there hasn't been lots of studies related to the influence of the shape on the magnetic properties. I'm not aware of any, so I don't know. But it's very interesting, should be addressed. Right, the size, of course, but the shape, yes. Because in this paper, which you know is about to appear, that I mentioned, where we have these hollow particles, nanoparticles. Well, we haven't done anything related to bio. It's just simple, you know, study of the magnetism and so on. So I'm not aware of it.
¿Podrías traducir, por favor?
Buenas tardes.
Okay.
El día de ayer ustedes hablaban de que las nanopartículas eran cubiertas con el medicamento para que ésta hiciera un efecto de taxi, de llevar el medicamento hacia el lugar que ustedes querían. ¿Ustedes han realizado pruebas de estabilidad en el tiempo de estas nanopartículas recubiertas? Es decir, uno de los retos de la industria farmacéutica es tener medicamentos que tengan una prolongación en el tiempo, que puedan ser conservados, ya sean a temperaturas bajas o a temperatura ambiente. ¿Ustedes han estudiado este efecto?
Yes. Yes, yes. Well, as I mentioned, we did look at the variation. Thank you for the question. We did look at the variation of sizes of these nanoparticles with time, you know, like 1 month and so on. And I think that I'm not sure if we looked into the shape, you know, the stability of it with time when you put the drug in. But it's something probably which has been assumed because the size hasn't changed and there's, you know, no reason why it should mature differently. And then the other thing is in these kind of tests, which is so far have been done only on mice, right? You kind of synthesize the thing. It's so easy to do. So you do it, you make a big batch and then you distribute it with time and so on. So it hasn't changed with time too much, but yeah, but this is something also very important to check in the future. Yes.
Hi. The last day, my question, another day. Yeah. Remember?
Yes.
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 Envía la pregunta, compañero. Envía, o sea, similarmente, analogous.
Yeah.
In the brain, what is the taxi for cancer cells in the brain? But because the neurons system nervous is difficulty llevarlas, o sea, following esas células cancerígenas.
Yes. Actually, you know, I haven't touched the brain because there is a huge program in Europe which is called the Brain Project, which is with the European Commission. And it's kind of like the funding in it is tremendous. So it's billions of dollars. And it's called the Brain. There are 2 huge projects now in Europe sponsored very generously, and all Europe, the best labs in Europe are working in it, like Germany, Italy, Norway, Sweden, England, all of these places, right? The best labs. And it's dedicated to brain. And the second one is dedicated to graphene, because of the importance of graphene. And the brain project is tremendous because there's many results coming out of it, and it's going at a quick rate. And in parallel, NSF and the US DOE, I think, and NIH, National Institute of Health in the US, have also selected the brain as one of their prime objectives. So they have been many top scientists are working on it. And they also included not only the science, natural sciences, as we call it, they also including social sciences. So it's a very rich field and it's moving quickly. And, I selected this thing about the brain because it enters into what we were discussing and so on. But this is just a small dot. So there's lots and lots of work on brain. Yes.
Okay, then we thank the Professor Marie-Louise.
Thank you.
Sorry, I don't recall the slide for the nanostructures. So when you were talking about maybe the bottom up, I guess, did you take advantage of the magnetic properties of these nanoparticles to build a bigger structure? Have you ever tried, or?
We haven't yet. No, but it's something that we should consider in the future. Yes. Yes. Thank you.
Okay. Thank you very much.
You.